Imagine seeing a woolly mammoth, not as a skeleton in a museum, but thundering across the Siberian tundra once again. Or what about the Tasmanian tiger, that ghost of a predator we've only ever seen in grainy black and white photos, resurrected into a living, breathing animal? This isn't science fiction anymore. Thanks to breakthrough science, we are on the verge of bringing these animals, and others, back from extinction. In fact, in 2025, the world met the first de-extinct animals created with this technology, 3 dire wolf pups. This process is called de-extinction, and it has officially moved from the pages of novels into the world's most advanced laboratories. It's a field driven by a powerful idea that we can actually reverse extinction. For decades, conservation has been about protecting what we have left. De-extinction proposes something radically different, a second chance. So you're asking yourself, how is this even possible? The answer lies in the building blocks of life itself, DNA. For species that have been gone for a long time, their cells are too degraded to clone directly the way Dolly the sheep was. That's where a revolutionary technology called CRISPR comes in. Think of CRISPR as a pair of molecular scissors. Scientists can now take the ancient DNA of an extinct animal pieced together from fragments found in old bones and preserved tissues and compare it to the DNA of its closest living relative. They then edit the living relative's DNA, snipping out and pasting in genes to match the genetic code of the extinct species. This edited DNA can then create an embryo, which is carried to term by a surrogate mother from the related species. The result isn't a 100% perfect clone, but a hybrid, a modern animal engineered to have the key traits of its extinct cousin. It's a scientific feat that was unimaginable just a generation ago. And a handful of iconic, long-lost creatures are at the center of this incredible new race. First on our list is probably the most famous Ice Age icon, the woolly mammoth. These giants, roughly the size of a modern Asian elephant, roamed a vast, cold grassland known as the Mammoth Steppe that stretched across North America and Eurasia. Covered in a thick, shaggy coat and armed with massive, curving tusks, they were perfectly adapted to a frozen world. Their ears were small to prevent frostbite, and they had a thick layer of fat for insulation. For thousands of years, they were a cornerstone of their ecosystem, shaping the landscape with their grazing. But around 10,000 years ago, as the climate warmed and human hunters got more sophisticated, the mammoths disappeared. The last isolated groups vanished from remote islands only about 40 years ago. Today, the company at the forefront of the de-extinction movement, Colossal Biosciences, is leading the charge to bring them back. Their plan relies on the mammoth's closest living relative, the Asian elephant. The two species share a common ancestor that lived about 6 million years ago, and their DNA is 99.6% identical. Led by renowned geneticist George Church, scientists at Colossal are using CRISPR to edit the DNA of Asian elephant cells. They're inserting mammoth genes responsible for key cold weather traits like shaggy hair, a thick layer of fat, and cold-resistant blood. In early 2025, as a proof of concept, the team announced they had successfully engineered mice to express some of these mammoth-like traits, like woolly fur, demonstrating their gene editing pipeline was working. The ultimate goal is to create a viable mammoth-like embryo. The challenges are enormous. Not only do they have to perfect the genetic edits, but they also have to figure out how to bring the calf into the world. Using an endangered Asian elephant as a surrogate is ethically tricky. So Colossal is also developing artificial wombs that could one day grow a mammoth embryo entirely outside a living body. Colossal has stated its ambitious goal of having the first mammoth hybrid calf born by 2028. The motivation isn't just to create a biological curiosity for a zoo. Proponents argue that reintroducing these giants could help restore the Arctic tundra. They believe mammoths would knock down trees and fertilize the soil, helping recreate the lost mammoth steppe ecosystem, which could insulate the permafrost and slow the release of greenhouse gases. But is this a brilliant ecological solution or a high-tech fantasy? Next up is a more recent and tragic victim of extinction, the thylacine, better known as the Tasmanian tiger. This unique animal wasn't a tiger at all. It was the world's largest carnivorous marsupial, a predator with the head of a wolf and the striped back that gave it that famous nickname. For thousands of years, the thylacine roamed mainland Australia, but competition with the dingo eventually confined the last populations to the island of Tasmania. When European settlers arrived, they saw the thylacine as a threat to their livestock. Fueled by fear and exaggeration, a bounty was placed on the animal's head. They were hunted relentlessly. The final chapter of this story is heartbreakingly specific. The last known thylacine, named Benjamin, died alone in a concrete enclosure at the Hobart Zoo on September 7, 1936, just 2 months after the species was granted official protection. But the thylacine's story might not be over. Colossal Biosciences is also working to resurrect this iconic Australian predator. Thanks to a well-preserved thylacine pup that was stored in alcohol for over a century, researchers have sequenced a nearly complete genome. This provides an incredibly detailed genetic blueprint for its revival. The plan involves editing the DNA of one of the thylacine's closest living relatives, a small, mouse-like marsupial called the fat-tailed dunnart. The team aims to create a thylacine embryo, and because marsupials have a very short gestation period, they are exploring technologies like artificial wombs and pouches to nurture the tiny embryo to maturity. Proponents argue that bringing back the thylacine could restore balance to Tasmanian ecosystems. As an apex predator, it would have preyed on sick and weak animals, a role that's been vacant since its extinction, possibly contributing to the spread of diseases like the facial tumor disease devastating Tasmanian devils. The science is advancing fast, and some believe a thylacine-like animal could be born within the next decade. Bringing back this ghost of the Australian bush would be a powerful symbol of our ability to undo past wrongs. Our third creature is one you may have never heard of, and its story offers a completely different path to de-extinction. Meet the quagga, a strange and beautiful subspecies of the plains zebra. Unlike modern zebras, the quagga had stripes only on the front half of its body. Its back half was a solid, horse-like brown, and its legs were pale and unstriped. These unique zebras once roamed South Africa in massive herds, but like the thylacine, they were hunted to extinction by European settlers who saw them as competition for their livestock's grazing land. The last wild quagga was shot in the 1870s, and the very last one on Earth died in an Amsterdam zoo in 1883. Her status as the last of her kind going completely unnoticed at the time. For over a century, the quagga was just a memory, but in the 1980s, DNA analysis of old skins revealed something astounding. The quagga wasn't a separate species of zebra, but a subspecies of the still-living plain zebra. This meant its unique genetic traits might still be floating around, just diluted within modern zebra herds. This discovery gave rise to the Quagga project, which began in 1987 in South Africa. Instead of high-tech gene editing, this project uses a much older method, selective breeding, also known as backbreeding. Scientists searched for plain zebras that showed quagga-like characteristics, like reduced striping on their hindquarters and a brownish body color. By selectively breeding these individuals over generations, the project aims to concentrate those dormant genes and produce animals that look and behave just like the original quagga. Today, after decades of work, the project has produced several generations of raw quaggas, which show a significant reduction in striping and increasingly resemble the extinct originals. These animals aren't perfect genetic copies, but they are a living, breathing testament to the idea that some extinctions might be partially reversible through careful, patient breeding. The science is incredible. The possibility of seeing a mammoth shake snow from its fur or a thylacine stalk the Tasmanian forests is truly mind-blowing. But this is where we have to hit pause and ask the biggest question of all. It's the question that echoes from the most famous cautionary tale about de-extinction, Jurassic Park. Your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should. Bringing these animals back opens a Pandora's box of ethical and ecological dilemmas. For one, what world are we bringing them back to? The mammoth steppe is gone, replaced by modern tundra and forests. An ecosystem isn't static, it evolves. Releasing a resurrected species into an environment that has changed for thousands of years could have unpredictable and disastrous consequences. Then there's the question of animal welfare. Cloning and genetic engineering are tough processes, often resulting in miscarriages, deformities, and chronic health issues. Is it ethical to create animals that may be doomed to suffer for the sake of scientific ambition? And what are we really creating? It's important to remember these aren't perfect resurrections. They are hybrids or proxies. A mammoth grown in an artificial womb and raised by humans will never be a true woolly mammoth. It won't have the social learning or instincts of its ancestors. Are we truly restoring nature, or just creating high-tech facsimiles? Perhaps the most potent argument against de-extinction comes from conservationists on the front lines of today's extinction crisis. They argue that the millions of dollars poured into resurrecting a few charismatic species would be far better spent saving the thousands of species currently on the brink. Why try to bring back the mammoth when we are struggling to save the elephant? It's a powerful point. However, proponents counter that de-extinction projects attract new funding and public attention, and that the technologies developed can also help save endangered species today. We've reached a point where, for the first time in history, we have the power not just to destroy species but to potentially bring them back. The revival of the woolly mammoth, the thylacine, and other lost creatures promises a future where we can correct some of our gravest mistakes. It offers the hope of restoring damaged ecosystems and reawakening a sense of wonder about the natural world. But this power comes with immense responsibility. We risk playing God, unleashing ecological chaos, and diverting precious resources from the urgent crisis of modern extinctions. The science is moving forward at an astonishing pace, but the ethical debate has only just begun. So, we turn the question over to you. Is de-extinction a revolutionary tool for conservation that we have a moral duty to pursue, or is it an act of hubris? a dangerous distraction from protecting the life that still shares our planet. Let us know what you think in the comments below.